Today we have Professor Julie Makani, who is the lead investigator for the Project 2 of the consortium. She's going to give us a presentation on the genomics of severe malaria in sickle cell disease. So, Julie, you have the floor.
Thank you very much. Greetings from Tanzania. I am going to share my slides. This is Project 2 of SickleGenAfrica consortium. And Project 1 is specifically looking at hemolysis. Project 3 is looking at the echocardiographic or echocardiovascular changes associated with hemolysis. And Project 2 is looking at malaria in association with hemolysis and sickle cell disease. So the three specific aims, or the specific sub-aims of Project 2, one, to characterize the phenotype of severe malaria in sickle cell disease patients, and to determine the relationship between variations in heme cytoprotective genes and malaria disease phenotype or severity. The second objective is to investigate the inflammatory response. This is looking at cytokines, chemokines, and adhesion molecules in sickle cell disease patients with malaria, and then to examine the relationship between the levels of these heme cytoprotective proteins and the risk of severe malaria. And then the third is to really functionally validate the association between the HCPs and the severity of malaria in transgenic sickle cell disease mice. For purposes of this webinar, I will just focus on the first project, the first specific aim. The second one is work that is led and is going to be conducted predominantly by Gordon, and we'll arrange another webinar to cover that. And then the third one with transgenic sickle cell mice and looking at severity of malaria in association with HCPs will be covered by Solomon in another webinar. So I'll just, going back to my, the bulk of the talk will be to look at the clinical phenotype or the phenotype of severe malaria. And just a bit of background, just to remind everybody, is that the burden of sickle cell disease is high, and the burden is highest in sub-Saharan Africa, with up to 80% of children who are born annually with SS disease born in Africa. Now, unfortunately, if you're born in Africa, your chances of childhood survival are low. And it's estimated that this ranges between 50 to 90% mortality if you're born and undiagnosed with sickle cell disease in Africa. Compared to other places, sorry, the projection of the slide is not very good. We need to address this. And one of the reasons, one of the factors that we need to address is malaria and the mortality that it causes in sickle cell disease. So if you look at this slide, you'll be able to see that newborn screening and managing infections has been able to reduce mortality, especially in the 0 to 3-year-old age group, by more than 70%. There has been very little change in the mortality above 5 years of age. So despite the various interventions, and this is particularly in the US, there's been very little change. I want us to focus on the under 5 mortality, because in our setting here in Africa, we still have infections, and malaria is a major cause of both morbidity and mortality in sickle cell disease. And this is why we felt that it's important to address this as one of the important projects in SickleGenAfrica. When you look at the geographical distribution of malaria and sickle gene, see that there is a very close overlap between malaria on the left side of your screen and the sickle gene. And this is because the presence of hemoglobin S, particularly in those in carriers, confers protection against severe forms of malaria and mortality due to malaria in individuals who are heterozygous for that. The irony or the tragedy is that despite the fact that individuals with sickle cell disease are protected from malaria, but when they do get malaria infection, it's associated with increased risk of morbidity and mortality. And this is why the recommendation is that individuals with sickle cell disease, SS disease, should be on malaria chemoprophylaxis, and any suspicion of malaria, there should be prompt diagnosis and treatment. Now, because we want to look at severe malaria, I really just wanted to give you a context of the rarity of severe malaria, but also this does not detract from the importance of malaria. Although it's common in our setting, and it's estimated that when you look at children, particularly under the age of 5, probably 50% of them are parasitemic each year, and they have fever episodes, and the probability of getting severe malaria is quite low, 3%, but it is associated with a high risk of mortality. And the question is, what happens in sickle cell disease? We're not really sure, because a lot of the work that has been done, the big trials looking at individuals have rightly so excluded individuals with sickle cell disease because of this high risk. And so we thought that as a consortium, we would be able to look and understand what happens in sickle cell disease, bearing in mind that it's absolutely important that we ensure that the care of sickle cell patients is not compromised in any way. I just want to reiterate the fact that in individuals with SS disease, despite the fact that there's a low prevalence of malaria parasitemia, it is associated with high mortality. And this is work that we were able to do in Tanzania, where we are able to demonstrate that the odds of malaria during hospitalization, or the odds of dying from malaria, if you're SS and you had malaria parasitemia, were fivefold. The odds were fivefold higher when you had malaria. The odds of mortality. And risk factors associated with malaria was anemia and a high AST. So the recommendation, this is a really important message, is that malaria in sickle cell disease is rare, how it is the serious cause of morbidity and mortality. And so the recommendation should be use insecticide-treated nets, make sure that you provide them with chemoprophylaxis, and then when you do detect it, prompt diagnosis and treatment with the recommendations, whether it's ACTs or quinellas, medications of severe malaria. So now I just want to talk a little bit about hemolysis in malaria and sickle cell disease, which is the underpinning question of SickleGenAfrica. When you look, this is a slide that shows red blood cells. These are normal red blood cells infected with parasites, and one of them has ruptured. And when you look at hemolysis in sickle cell disease compared to hemolysis in malaria, in that in both instances, we're not really sure if you have malaria and sickle cell disease, whether this is a complication of malaria or this is a complication of sickle cell disease, because there's quite a lot of overlap with some of the clinical presentations that you see. So in both instances, you have anemia. However, in the mechanisms or the the pathogenesis of anemia may differ. In sickle cell disease, you may have cerebrovascular accidents, and in severe malaria, this may be associated with cerebral malaria. And then in some individuals with sickle cell disease, when they get acute chest crisis, it may be difficult to distinguish this from the acute respiratory distress syndrome that has been described and is a definition of severe malaria in particularly in children. So just a little bit about heme cytoprotective protein. This is the interest of SickleGenAfrica. There are three main mechanisms. These are proteins that help in reducing the dangerous or the deleterious effects of cell-free hemoglobin, free heme, and iron. And you have haptoglobin, hemopexin, alpha-1 microglobin, which really scavenge and remove danger molecules. You have hemoxygenase 1 or HO, and then you have ferritin. The hypothesis that we wanted to explore, the one that we want to investigate, is that when you look at the hemolysis associated factors, how are they influencing and how are they associated with malaria, the presence of malaria, and how are they associated with the presence of severe malaria? And we decided to focus on three of the main causes, or I mean the main types of severe malaria that I'll talk to later. So the question is how to describe what happens, how do we look at the association, and then how do we determine and how do we look at the association of the genetic factors or the genomic variants that may be associated with malaria, HCPs, as well as sickle cell disease. And so these are the main questions. And this, again, remember, this is just for the sub-aim 1 for project 2, is the presentation of severe malaria and sickle cell disease. What is the inflammatory response to malaria, and what is the mechanism of disease, looking at transgenic mouse models. So this is you see the project here. And essentially what project 2 is about is, first of all, is to enroll 6,500 individuals with sickle cell disease, 2,000 from Tanzania, 2,500 from Ghana, and 2,000 from Nigeria in different sites. Enroll these patients so that we can have a clear description of the association between HCP and malaria or no malaria during steady state. And so it will be a cross-sectional study done at clinic where these patients are being enrolled. And then we will follow patients up and look at them during hospitalization. And the aim is that for the 3 countries, enroll 500 patients each who are hospitalized and look at the prevalence of malaria in during the acute stage. And based on the studies that have been done, we estimate that the prevalence of malaria at clinic is 1%, and the prevalence of malaria during hospitalization is 5%. So it's very rare, and this is why it's really critical, and it's great that we'll be able to look at the numbers across the sites, because it would be very difficult for one to answer this question just from looking at one site, because malaria is so rare. The prevalence of severe malaria is even more rare. It's 0.5%. And so the phenotypes we're interested in are really phenotypes that, as I said, are both in sickle cell disease and associated with malaria. So when you look on the left-hand side of the screen, the neurological complications in sickle cell disease, stroke is a known complications, seizures, and loss of consciousness. When you compare this with severe malaria, cerebral malaria is also associated, or also has been described to present with stroke, seizures, and loss of consciousness. So the question here is that when we're looking at the phenotypes and we're looking at it, will we be able to say, in those who are, have malaria, how do we distinguish whether this is a complication due to disease or due to sickle cell disease itself or due to malaria? And then when it comes to cardiopulmonary complications, in sickle cell disease, they get acute chest syndrome associated with low peripheral oxygen saturation of less than 95%, associated with new pulmonary impacts, and then acute respiratory distress syndrome, which is characterized by chest indrawing and also an element of hypoxia. Severe malaria or anemia is a recognized cause of severe anemia, or is a recognized complication of severe malaria. And we also have that in sickle cell disease. And in both instances, there is hemolysis. And then one thing, of course, that doesn't happen in malaria, although you do have elements of myalgia, this is more of a mild pain. But in sickle cell disease, the hallmark of the disease is severe painful episodes. So what we want to do is, in both steady state as well as during the acute stage, we want to be able to describe these phenotypes so that we have a very accurate description of disease, so that we can then relate it to malaria and HCPs. So just to repeat the numbers again, at steady state, we'll be looking at 6,500 individuals across 3 countries. During the acute stage, we'll be looking at, sorry, this is an error. We'll be looking at 1,500 individuals, not 1,000. And then the expectation is that at steady state, 1% of the individuals will have malaria, and 5% will have severe malaria during hospitalization. So the study design, the first one is really a longitudinal study or a cohort study where we just enroll 6,500 individuals and look at what happens at steady state. And then the longitudinal studies will be more of a case control study. The cases will be sickle cell patients who are hospitalized at least one or more times with severe malaria during the study period. And then we have two types of control groups. There are those sickle cell patients who during the study period will never have malaria at all. And then, the second control group will be those who have malaria but do not develop severe malaria. So these will be patients with mild, moderate, or what is called uncomplicated malaria. In terms of looking at the disease modifiers, the factors that we'll look at to determine association is we'll be comparing, in the first instance, we'll do an analysis where we'll compare individuals with sickle cell disease and severe malaria against individuals with sickle cell disease with severe malaria or with mild malaria. And so we'll look at both the clinical laboratory factors that are associated with severe malaria and uncomplicated malaria. And this would include looking at the genomic variants that have been described to be associated with HCPs and look at the association and whether there's any factors that can be found to be either increased or decreased in one or the other group. The second comparison will really be a simple comparison where we'll be taking the individuals with malaria, irrespective whether they have signs of severe malaria or not. We'll just compare those with malaria and those without malaria. And the question here that we want to look at, or the association that we want to look at, is that what are the factors that are associated with the risk of malaria infection? And again, this would include genetic variants. So we expect that at the end, we will be able to, first of all, have a description of malaria and sickle cell disease in a large population, and in a population that's not only large, but that is scattered across Tanzania, Ghana, and Nigeria, which will be very novel because this is something that hasn't been done before as part of a multi-site study. And we hope that with the spread and with the geographical distribution, as well as with the numbers, we'll be able to provide additional information with regards to malaria and the forms of malaria in sickle cell disease. The second thing that we'll be able to do is that we'll be able to look at very specifically the disease modifiers related to HCPs, which are associated, one, with just the presence or absence of malaria infection, and then also look at the factors that are associated with severe malaria. So in this instance, when we look and identify the small group of individuals who do develop or who will develop severe malaria. And then, of course, looking at the genomic variants which are associated with HCP and malaria in sickle cell disease. So with this information that we have, this will also be able to inform and complement the two other specific aims of Project 2, which is the project on malaria. So they'll be looking at inflammatory responses, and they'll be able to look and test some of the findings that we have found in patients in transgenic sickle cell mice. So the conclusion is that with Malaria 2, very specifically Project 2 and Sub-Aim 1, we will, at the end of the day, have a description of the prevalence, pattern, and outcomes of malaria and sickle cell disease. We'll be able to contribute to understanding the hemolysis that occurs in malaria and sickle cell disease and how this feeds into each other and feeds into either worsening or influencing the natural history or the occurrence of disease, both malaria and sickle cell disease. And also, just as important is that we'll be able to improve the management of both hemolysis, malaria, and sickle cell disease, both in the acute stage, and hopefully we'll also be able to get some insight into end organ damage. The aim really is to use the information that we're able to collect and the knowledge that we're able to generate from SickleGenAfrica to inform treatments, to inform understanding, and to contribute as scientists from Africa contribute to genomic research that could potentially catalyze discoveries in Africa that would also be of value all over Africa. So thank you very much. This reaches the end, and because we've got quite a lot of time, I'd be happy to take as many questions as possible, or to highlight some of the questions or some of the slides that have been projected. Thank you very much.
Thank you very much, Julie. Let me set the ball rolling with my question. So the question is, have you made any progress? Where are you in terms of initiating Project 2?
So at the moment, year one was involved with the enrollment of patients into the into SickleGen registry setting up and everything, and the completion of year of the first year of enrollment. Across the sites, we've enrolled at least 2,000 individuals into the SickleGen Africa registry. At the moment, we're now finalizing the CRFs so that we can then start collecting the information on Malaria Project 2. We're hoping that at the consortium meeting next week in Tunisia, we'll be able to have a side meeting that we can finalize the CRFs, and the aim would really be beginning of May to start actively enrollment of at least the acute phases or the hospitalized cases of malaria. So that's the status of where we are.
Thank you.
Any other question, comments, suggestion, thoughts? Maybe just to acknowledge one of the limitations is that despite the fact that this will be a large study with 6,000, I think it will be probably one, if I'm not mistaken, the largest study of sickle cell disease looking at malaria with 6,500 individuals. When you look at the rarity, and it's something to be thankful for. When you look at how rare sickle cell disease is, I mean malaria is, we still will have quite a small number of individuals who will have malaria and who will have severe forms of malaria. And so we will be looking at small numbers of the cases of interest. Any questions or comments or thoughts?
So my other question is, where are you going to genotype your samples eventually?
So, and maybe David or can also comment, but all the genotyping for SickleGenAfrica is going to be centralized, and the genotyping is going to be done in South Africa, in Cape Town. And if there are any additional genotyping that will need to be done, this can be arranged separately. But currently, all the samples will be collated from the sites, sent to Nigeria, which is where the biobank is, and then the genotyping will be done in CPGR in Cape Town.
Oh, any more questions? It doesn't look like that. Well, so if there are no further questions, then we'd like to thank Julie for this brilliant presentation, as always, and in the meeting. So we meet again next month. Thank you. Bye.
Thank you very much. Bye-bye, Anita, and bye-bye, everyone. Thank you for making the time to join.